Hello and welcome to Cognito Key. Today, we're diving deep into a world you can't see to uncover a biological puzzle that really messes with our definition of life. We are talking about something truly strange. We're talking about viroids. So, let me start you off with a little riddle. What do you think is smaller than a virus and is an absolute master of disguise inside the plant world? Now, the answer? It's not a tiny bacteria or some simple virus. Nope. It's something way, way weirder. The answer is the viroid. And what is it? Well, at its heart, it's basically nothing. It's just this tiny, circular loop of RNA. And the most important part is that it's naked. You see, unlike a virus, it has no protective protein coat. This really bizarre agent was first figured out and given its name back in 1971 by a plant pathologist named Theodor Diener. But the story of this mystery actually starts way earlier. Scientists had noticed these odd plant diseases all the way back in 1917. But the real breakthrough? Well, that took a while. It wasn't until 1967 that Diener and his colleague Raymer pinpointed the culprit behind a disease that was just wiping out potato crops. And what was it? The potato spindle tuber viroid. This was huge. It proved that an infectious agent even smaller than a virus could exist, which led Diener to officially coin the term viroid in 1971. You know, the easiest way to wrap your head around a viroid is to just compare it to a virus. So viruses are basically genetic material, could be DNA or RNA, all wrapped up in a nice protein coat. Viroids, they just tear up that whole rule book. They are only RNA. That's it. No protein coat, and get this, no instructions to even build proteins. They are the absolute ultimate in biological minimalism. So let's get into it. What are the key features of this master of simplicity? Let's break down the viroid profile. Okay, so viroids are, quite simply, the smallest infectious agents we know of. Their entire being is just a single, stable loop of RNA. This incredible simplicity is why some scientists think they're molecular fossils, like a little leftover from an ancient RNA world, from a time long before DNA took over. And their whole strategy? Pure hijacking. They just force a host cell to make copies of them, and they cause disease by messing with the plant's own genes. It's a process called RNA silencing. Basically, they turn the plant's own defenses against itself. Pretty sneaky, right? Now, even with all this simplicity, viroids aren't all the same. They actually fall into two main families, and what separates them is really simple. It's all about where they decide to set up shop inside the host cell. And the divide is pretty clear. About 70% of them belong to the Popsiviroidae family, and they replicate in the cell's nucleus. That's kind of like the cell's command center. The other 30%, the Avsunviroidae, they take over the chloroplasts, which are like the plant's little solar power factories. It's a simple difference, but it completely changes how they go about making copies of themselves.
Which brings us to the big question, right? How on earth does a single loop of RNA, with no genes of its own, trick a whole complex cell into making copies of it? Well, the answer is a brilliant little strategy of cellular hijacking.
This strategy is called the rolling circle mechanism. Just imagine the viroid's RNA loop is like a circular mold. The cell gets tricked into using this mold to just print out these long, continuous ribbons of copies. And this whole process has two distinct flavors, one for each of the viroid families. So first up is the asymmetric method. This is what the family that replicates in the nucleus uses. The host's own enzymes use the original viroid as a template to create a long, complementary negative strand. Think of it like a film negative. Then another host enzyme comes along and acts like a pair of scissors, cutting this long ribbon into individual units. Finally, these little pieces are stitched into perfect circles, and voila, new infectious viroids are born. Now, the second family uses a symmetric method, but with a really clever twist. The viroid RNA has its own built-in scissors. It's a special kind of RNA called a ribozyme, and it can actually cut itself. So after the first copy gets made, the viroid just snips itself free. This new piece then becomes the template for the final copies, which also cut themselves free before getting stitched into new viroids. It's a much more self-sufficient, do-it-yourself kind of process.
This totally unique biology leads us to a really powerful idea. This quote here says it all. Viroids are so much more than just plant pathogens. A lot of scientists see them as a window into the distant past, living relics from that ancient RNA world that existed long before DNA and proteins ever came on the scene. In a way, they are living history.
And all of this leaves us with a final, absolutely fascinating question. Sure, we study viroids to protect our crops, But as we untangle their brilliant, minimalist survival strategies, you just have to wonder, what deeper secrets do they hold? What can these tiny, naked loops of RNA really tell us about how life on this planet even began?
We really hope you enjoyed this little journey into the amazing world of viroids. If you found this explainer interesting, please do give it a like and share it with your friends. And of course, make sure to subscribe to Cognito Key for more deep dives into the world all around us. Thanks for watching.